Hello there! I would like to discuss about the parts of the microscope. But before we get there, you should know what a microscope is and also a part of its history. What is the microscope? For millennia, the smallest thing humans could see was about as wide as a human hair. When the microscope was invented around 1590s, humans suddenly saw a new world of living things in the water, in the food, and under their noses. Microscopes are instruments that use lenses and light to enlarge the image of the object or specimen placed on the slide. For example, on the left is a picture of blood smeared onto a slide. On the right is a microscopic view of it. Every major field of science has benefited from the use of some form of microscope, an invention that dates back to the late 16th century and invented by a modest Dutch eyeglass maker named Zacharias Janssen. While extremely rough in image quality and magnification compared to modern versions, the Johnson microscope was nonetheless an influential advance in scientific instrumentation. Janssen was the son of a spectacle maker named Hans Janssen, and while Zacharias is credited with inventing the compound microscope, most historians assume that his father must have played a vital role since Zacharias was still in his teens in the 1590s. In fact, some historians credit both the Janssens and a fellow Dutch eyeglass maker, Hans Lippershey, with concurrent, though independent, invention of the microscope. The device was described as an object that rose vertically from a brass tripod almost 2.5 feet long, as you can see in the picture. The main tube was an inch or two in diameter and contained an ebony disc at its base, with a concave lens at one end and a convex lens on the other. The combination of lenses enabled the instrument to bend light and enlarge images between 3 and 9 times the size of the original specimen. Very different from the microscope we see in science laboratories nowadays. Now let's move on to the parts of a microscope. There are 3 structural parts of the microscope: the head, base, and arm. The head. This is also known as the body. It carries the optical parts in the upper part of the microscope. Next is the arms, or arm, the carrying handle. This is the part connecting the base end to the head and the eyepiece tube to the base of the microscope. It gives support to the head of the microscope, and it also used when carrying the microscope. Next is the base. It acts as the microscope support. It also carries the microscopic illuminators. The optical parts of the microscope are used to view, magnify, and produce an image from a specimen placed on a slide. These parts include: the first is the eyepiece, also known as ocular eyepiece. This is the part used to look through the microscope. It's found at the top of the microscope, and its standard magnification is 10 times, with an optional eyepiece having magnifications from 5 times to 30 times. The second is the eyepiece tube. It is the eyepiece holder. It carries the eyepiece just above the objective lens. Next is the objective lenses. These are the major lenses used for specimen visualization. They have a magnification power of about 40 times to 100 times. There are 1 to 4 objective lenses placed on one microscope. Each lens has its own magnification power. A scanning objective lens provides the lowest magnification power of all objective lenses. It has a magnification power of 4 times. The low power objective covers a small part of the objective lens, but in terms of the field of view, it is wide, and they are useful for examining large specimens or surveying many smaller specimens having 10 times power. The high-powered objective lens is ideal for observing fine details within a specimen sample. It has a magnification power of 40 times. Imagine! And the last one is the oil immersion objective lens. It provides the most powerful magnification of 100 times. A drop of special immersion oil is added because if not, the specimen will appear blurry. Next one will be the nose piece. It is also known as the revolving turret. It holds the objective lenses. It is movable, hence it can revolve the objective lenses depending on the magnification power of the lens. Next one is the adjustment knobs. These are knobs that are used to focus on the microscope. This one, as you can see in the picture. There are 2 types of adjustment knobs, the fine adjustment knobs and the coarse adjustment knobs. The coarse adjustment is used to focus the microscope. It is always used first, and it is used only with a low power objective. Next one is the fine adjustment. It is used to focus the microscope and is used with a high power objective to bring the specimen into better focus. The next part will be the stage. This is the section on which the specimen is placed for viewing. They have stage clips, the smaller ones here. The stage clips hold the specimen slides in place. The most common stage is a mechanical stage, as you can see here in the picture, which allows the control of the slides by moving the slides using the mechanical knobs on the stage instead of moving it manually. The next one will be the aperture. This is a hole on the microscope stage, this one, through which the transmitted light from the source reaches the stage. The 9th part is the illuminator, or the microscopic illuminator. This is the microscope light source, located at the base. It is used instead of a mirror. It captures light from an external source of a low voltage of about 100 volts. If your microscope has a mirror, it is used to reflect light from an external light source up through the bottom of the stage. Next part is the condenser. It is used to collect and focus the light from the illuminator onto the specimen. They are found under the stage next to the diaphragm of the microscope. They play a major role in ensuring clear, sharp images, which are produced with a high magnification of 400 times and above. The next one is the diaphragm. It is also known as the iris. It is found under the stage of the microscope, and its primary role is to control the amount of light that reaches the specimen. It's an adjustable apparatus, hence controlling the light intensity and the size of the beam light that gets to the specimen. The next one is the condenser focus knob. This is a knob that moves the condenser up or down, thus controlling the focus of light on the specimen. The microscope is the most influential technology that allowed us to study and observe things that we could not be able to see with the naked eye. The microscope helps to identify viruses and diseases. These discoveries led to preventative measures such as vaccines. Johnson was the first to realize that by inverting the lens structure of early prototype telescopes, we could get a higher degree of magnification. The initial microscopes were simple compound types, combining a magnifying objective lens with an eye lens. Advancements soon followed through, and after Robert Hooke published Micrographia in 1655, the microscope became a staple tool for any scientist. So that's all for this topic. Thank you.